Thank you very much for the introduction and also for inviting me. It's a real pleasure to be in San Francisco. San Francisco is one of the cities to be considered back home, and I keep sending them messages about the beautiful weather and about the very cheap wine. In fact, California wine in the Netherlands is half the price when compared to San Francisco. Today I will discuss mushroom-forming fungi converting plant waste streams into novel biomaterials. And before doing that, I would like to introduce our research line on mushroom development. In the Netherlands, we produce 300 million kilograms of white button mushrooms every year, making the Netherlands the second largest producer of these mushrooms in Europe. And it's a high-tech industry, but nobody actually knows what the molecular mechanisms are that underlie the development of these fruiting bodies. And we study this by using the split-kill fungus as a model system. And we can find Schizophilum on all continents, also in California. We have a large natural collection of 100 strains from all places we visit during our holidays. Like we have a strain from the Kremlin in Moscow, and the Chinese Wall, and the palace of the Japanese emperor, and more of those kind of places. And this fungus can establish its life cycle in 1 week on defined medium, both on Petri dishes and even in 96 well plates. There's a 15% genetic diversity between strains. That is the same as between man and cow and man and cat. So it's enormous. And it has 13,000 genes, as was shown by JGI sequencing project. And we recently showed that there are even 15,000 transcripts due to a highly program of alternative RNA splicing. Molecular tools are also available. So this fungus first establishes, establishes a fertile dikaryon by fusion of two compatible monokaryons. And when the environmental conditions are favorable, fruiting body development can be initiated, meaning that first the fungus starts to grow asymmetrically, then it forms aggregates that further develop in primordia, and these primordia can then grow out into mature mushrooms. And we started a program to delete all 450 transcription factors. We are only a few percent because we only have like 10 deletion strains, but in the end we would like to inactivate them all. And this is one of the examples. This is a homeodomain protein, and what you can see here, and can I go back? Yeah. What you can see here is the wild type forming a ring of mushrooms, and this is the deletion strain of HOM2. You can see there's no asymmetrical growth and there's no fruiting bodies formed. And very interestingly, and I will come back to this later, is that this phenotype is accompanied by an increased growth of the vegetative mycelium. We can see a twofold higher biomass formation if this gene is absent. So we get induction of mushroom production and at the same time a reduction of vegetative biomass at the moment this gene is present. And even when we combine two deletion strains, we even have a threefold increase in biomass of the vegetative phase. Now, we made a constitutive version of this homeodomain gene, and what you can see that in the dark and at high CO2, we can see that hardly the fungus is growing, and when we transfer this to light and ambient CO2, we can see immediate formation of mushrooms. Another example is the C2H2 deletion strain. This strain grows asymmetrically like the wild type. It forms aggregates, but primordia and mushrooms are not being produced. And this made us the first model of the formation of mushrooms, and this model based on the split kill fungus also has predictive value for other mushrooms, as is shown in the next slide, where we overexpressed C2H2 in the white button mushroom. And it's a complicated figure, but what is shown over here that we had a 1 day earlier mushroom production when compared to the wild type, meaning that this strain produced mushrooms in 20 days, whereas the wild type takes 3 weeks. And based on the sales of white button mushrooms, such a strain would yield 100 million euros in Europe on a yearly basis. And we had a patent on this. We tried to sell it to the companies, and nobody wanted it, not even the American Sylvan company. And this makes it I'm still here and not financially dependent. So the next step is that we are trying to do another commercial project, and that is a project on using the vegetative mycelium And we should acknowledge the fact that the mushrooms, as we see them in the forests, are only a minor part of the body of the fungus. 95% or more is even in the soil. And there are mushroom-forming fungi, and it was already mentioned in the previous talk, that can colonize surface areas like 10 kilometers square. And we will make use of this to make new materials. So what we do is we inoculate a waste stream, like straw, wheat straw, or sawdust, with the fungus. And the fungus starts to colonize on it, and part of the material is being digested. And while growing, all these hyphae bind together the wheat, the straw fibers. And by doing this, the fungus is gluing all these particles together. So after colonization, we can add the colonized material to a mold, and we can start pressing it. And we put 40,000 kilos on top of it, and after that we can take it out, and we have made a hardboard-like material. And in contrast to the commercial hardboard you buy in the shop, there's no involvement of formaldehyde or epoxy. It's just a waste stream combined with fungus, pure natural. So we were actually not the first that started with these kind of projects. It was MycoWorks and Ecovative, both based in the US, that started to explore the use of fungal materials. And for instance, Ecovative launched this insulation material that is able to replace glass wool or rock wool-like materials. Or they started to create packaging materials which should replace plastic packaging materials. And companies like Dell and IKEA announced that they wanted to replace their plastic packaging materials for these kind of fungal-based materials. And there were some cynical people that mentioned that this was kind of a greenwashing program. And I prefer to be naive, but still there is no replacement yet of these kind of materials. The plastics are simply too cheap at this moment. So we can also think of some high-end materials like Phil Ross and Maurizio Montalti, who have designed furniture based on mycelial materials. The good thing is that you can grow any form, and another appealing application are the plant pots, as shown over here. Plant pots in the Netherlands are very important. The Netherlands represents the main center of horticulture in the world, and the Netherlands uses 3 billion plastic plant pots a year, and they are all put in the garbage. So if we could replace those pots for fungal material based pots, that would be great. The price of our pots is, however, a little bit more expensive, but if we can add functions that plastic do not offer, then they may be competitive. For instance, if these pots would supply us with fertilizer for the plants, then we may be able to replace the plastic pots for these kinds of sustainable alternatives. So it was also Phil Ross that started to make bricks out of fungal materials, and we are close to the European Space Agency. That is, the research center is based in the Netherlands, and they approached us whether we would be willing to collaborate with them to see whether we can make bricks for building on the moon. And the idea is that we cannot transport bricks by space ships to the moon or to Mars, and also mining at those sites is too expensive. So the idea is to bring a plant to the moon, and we chosen the fastest growing plant in the world, that is Azolla, a water fern, and that can fix both nitrogen and CO2. And that would be grown, and then we add a little bit of fungus to it, and the fungus will colonize the plant material and make a solid structure, and a robot would then build this kind of house, and then later on we can go to the moon and live in these kind of houses. And we have started to do some 3D printing experiments, and actually that works. This is a plant paste of Azolla. We have added the fungus to it, and the fungus will fortify this structure. So far what we did was we inoculated a waste stream with the fungus. We let it colonize for 3 to 4 to 5 to 6 days. It's up to us to decide. And after that period, we inactivate the fungus by a heat treatment. The fungus is killed. And then we get what we call a composite material, which is about 95% plant material and 5% fungal material binding these plant fibers together. What we could also do is we can have this plant material degraded completely, and then we end up with a pure mycelium. And if we look under the electron microscope, pure mycelium has a ultrastructure very similar to that of a textile. And that made us to produce the first fungal dress. And what you can see over here, you can see the petri dish forms, because we grow all these mycelia in petri dishes. And it was Aniela Hoiting who has made this dress, and she is a designer for Tommy Hilfiger. Through the week, and at Friday and Saturday she spends time on producing new kinds of textiles, and she collaborates with us. And this in fact attracted world attention. We were in a very serious journal in England, the Daily Mail Online, and we were actually next to Kim Kardashian. And that made my daughter, for the first time, to have some respect for me. Because she thought, if you are next to her, then your work must be important. So, and the dress was also in a fashion show in the Netherlands, and so you can really wear it. There are now 3 dresses. This is the one on the fashion show. This is Aniela, and the nice and good thing is that the first dress you could not touch, but the third one you can already wear. So we make real improvements. Here's the model with the dress on, so it's nice. So Mycelium textile has really additive functions because, first of all, it's sustainable. You have to realize that a white cotton shirt, as I'm wearing at this moment, costs 2,400 liters of water to produce. And this jeans here takes 7,000 liters to produce. And with the climate change, more and more areas get drier and drier, jeopardizing cotton production. And in our case, our Mycelium dress only took 12 liters to produce. So that is a huge improvement, but I must say that we did not dye the fabric. We can do it. It will increase the water usage, but still it's much better than producing cotton. At the same time, these kinds of materials, they also contain bioactive compounds like this molecule. This is schizophyllin. It's a beta-1,3, beta-1,6 glucan. And this promotes epidermal renewal and protects against photoaging. And that is why these kinds of polysaccharides are already used by the industry and added to daycare products. So while wearing our dress, your skin is nurtured. Another thing is that it is self-repairing. So if cuts like this is when they are out of fashion, we can simply repair it. You get a bottle of fungus with it. It will grow overnight, and you can iron to inactivate the fungus, and it is repaired. Also, when you grow in weight, you can extend your shirt. It's all possible. So what we do at the moment is to create a palette of mycelium materials by varying the type of waste stream, the type of fungus. We also use genetic modification by varying growth conditions and post-treatments. So what we see here are 2 things. First, if we grow the fungus in the dark or in the light at low or high CO2, makes that we get different types of materials with different properties. And what we also see, and I don't have time to go into this, but we have a deletion strain called delta SE3, and this is deleted in a hydrophobin gene. And to our big surprise, what you can see is that this material is 3 times more strong when compared to the wild type. So genetic modification indeed has a huge potential in creating novel materials. So the normal fungal materials are in the range of the what we call natural materials like wood and paper and cork, whereas the deletion strain, as you can see, has moved to a different class of polymers. Those are the plastics like the polyethylene and the polyphenol chloride materials. So we really have developed now plastic-like materials, and we would also like to continue to produce metal-like materials, elastomer and foam-like materials. So what is the basis of the fact that this deletion strain is a more strong material? It can be either because of an altered cell wall structure, composition, or the density of the mycelium has increased. Well, we did some infrared spectroscopy studies and we found an identical cell wall composition. So that was not the explanation. The explanation indeed was that the density of the material of the deletion strain is much higher when compared to the wild type. And this is shown also by the scanning electron microscopy. Here you can see the wild type, it's a fluffy-like material, whereas the deletion strain is very much more dense when compared to the wild type. So is it possible to create materials with a different cell wall composition? Yes, it is. This was sent to me last night actually, and we for the first time have now a schizophyllum. By growing not under wet conditions but on dry conditions, we see now for the first time an acetylated form of the glucans that are contained in the cell wall. And this is interesting because it's known that acetylated glucan has different properties. For instance, in this case it was shown with increased cohesiveness, springiness and gumminess. So we may have now obtained a novel material and we have to further characterize it. Also, we will focus on the growth speed. We have these deletion strains that were originating from our mushroom program that showed increased growth. Increased growth means faster product formation, and we will look at the properties of these strains in the near future. So this brings me to the acknowledgments. We are a biology group and we need material scientists. So we collaborate with the Technical University in Delft. We need nice products, so we collaborate with the Design Academy in Eindhoven and with several designers. And most intensely we collaborate with Maurizio Montalti, and Freek Appels and Paulien Krijgsheld did most of the work. Joint Genome Institute sequenced the split-kill fungus and now will sequence more and more strains of this fungus, and we are funded by the European Space Agency and the Dutch Science Council. And I would like to end with the most beautiful mushroom, the wood ear, and I would like to thank you for listening.